So we will finish this chapter. We just have a couple of slides, and we will finish that, and then we will do a review. And what you will see that I actually posted like a PDF file yesterday about exam review, and a lot of people actually just asked like, okay, all right, this is not a study guide. So this is just list the topic that we need to cover. Believe me, there are some people, there are probably a few probably still don't have the textbook. So even for them, that is helpful. So we will go a little bit beyond that, and I actually uploaded a PowerPoint file which actually does have some material, and then we're going to talk about that. But let's first finish that, and there are some other issues that we'll discuss that. So we finished this part, but I definitely wanted to stop by to this one because a few of you were confused about the material, what is in here, and it's absolutely legit. I mean, like, there are some confusing parts in here, and you will see that there is a whole paragraph under this about this transepithelial transport in the intestinal wall, as well as the parietal cells, which will be the next slide, making the doing the acidification of the stomach, and definitely read those two materials. So the first thing that what we are looking here, that when you are, these are the cells, the apical surface is towards the intestinal lumen. Then you have the basolateral surface. This is towards the connective tissue, which does have the blood supply. So the difference in here is a lot of people actually thought, okay, all right, so here you are having your meal, your glucose is getting broken down from your complex carbohydrate to glucose, many other monosaccharides, of course, but we're just taking a look with this. So you have this glucose right here, and you are transporting that glucose using a symporter, where sodium is driven down the concentration gradient. Sodium is always outside. The cell is more. Inside the cell is less. This is universal truth. So sodium is going down the concentration gradient and taking the glucose up the concentration gradient. That's the important part. So glucose is more here. Now, immediately the question that you will ask if you have studied the material, that when we were talking about the uniporter of the glucose, and we can just go by just quickly to just revisit that what I'm talking about here. If you actually look right here, this one doesn't make sense here, because I already said that once the glucose goes into the cell, so that the glucose transport always goes in one direction in the uniporter, it is changed into glucose 6-phosphate. So this is the reason that glucose is going to go into the cell and following this uniporter, which is going from a concentration of high to low. And here, if you just look, the scenario that what we are talking about right now is entirely different. Okay. Right here is entirely different. So what is the difference between that picture and this picture? Anyone? Small difference. Which one? Yeah, that's correct. That's very important. The previous one, the example of GLUT1 transporter, very different Km. Here you have an entirely different transporter, which is GLUT2. And the physiology of these two types of cell, what is the representation of the last one and this one, is very different. So that's the reason that it gets confusing. So what is happening, that the previous picture is a generalized view of all your body cells that requires glucose for their functioning. This could be your skin cell, muscle cell, heart cell, neurons, any other cell in your body. It needs the glucose from outside. It keeps each and every molecule available inside, transfers into glucose 6-phosphate. That will go into the glycolysis, then Krebs cycle electron transport change to produce energy for that cell. Here the function is entirely different. You have a huge amount of glucose coming in after the meal. That cell will not be able to keep all the glucose because it is way too much for it. So it will actually not convert except a few molecules to glucose 6-phosphate, which is just needed for the functioning of the cell. Rest, it will remain as glucose to be transported outside of the cell to go and it will go to the rest part of your body. So that's the main difference between all your physiological cell all throughout the body. And this is a temporary warehouse for the glucose, and all the glucose is not going to get transformed into the glucose 6-phosphate. So as glucose is being transported and stored in glucose form inside the cell, the glucose concentration is going to go high. And that's the reason that, say, the glucose concentration is higher here as it is constantly being transported. And that's why you need a symporter to go uphill, going from low concentration of the glucose to high concentration of glucose. And that glucose now being in a high concentration, GLUT2 have actually a low affinity for glucose. So it will not work like a GLUT1, which has a very high affinity for glucose. GLUT2 will work perfectly here because you have a very high glucose concentration inside the cell. So GLUT2 will perfectly work. The more glucose in this cell coming in, the more faster the GLUT2 will actually take it away to the body cells. So essentially, not really the body cells. The way it works, that it will go to the blood and your insulin will rise, and mainly the liver cells will take up the glucose again by a GLUT2. And that you do not have to go into that part, but it will go to the liver, converted into the glycogen. And when it's needed, that will be delivered to all your body, all your other cells in your body for their own function to produce energy. And these will be the GLUT1 transporter, which is a uniporter with a very high affinity of glucose. So 5 millimolar glucose concentration normally in blood, that is good enough for these GLUT1 transporter to transport glucose in here. With a 5 millimolar blood glucose, that GLUT2 will not transport. You need a much more higher value for that. So that's the main reason that why the glucose is not converted to glucose 6-phosphate, because this is a warehouse for the glucose. It is being transported elsewhere in the body for the rest of the part of your body to work. So, and again, what are the different? So you have a symporter, you have the GLUT2 uniporter, different type with a different Km than GLUT1. And of course, you have the sodium-potassium ATPase, because the sodium is coming in. And for the sodium to come in, you have to have a low concentration of sodium inside. So those sodium need to be pumped out in exchange of the potassium to maintain the sodium gradient. And that excess amount of potassium is also going to go out to maintain an equilibrium of potassium. And this is your potassium leak channels. So you have a channel, you have a pump, you have an uniporter, you have a symporter. So again, and that actually maintain all the, see that low sodium, high potassium, high sodium, low potassium. And here you can see that high dietary sodium and the chloride as well. So here there's like an extension of the material. What happens that as sodium and glucose is coming into the cell, it actually brings water along with that. And that water does not move through the cell through aquaportin. So that's an interesting part. That's an extra part. I'm not going to ask any specific questions, but you will see that it is after the section that you have in your textbook. Most of the water actually moves from the side, like through these tight junctions. It's not going to enter the cell, but the gap that you have in between the cell, that is enough for the glucose to move, which is called as a paracellular transport. So that's how your water is actually taken up. So this is actually the important thing, that when you want to rehydrate someone, it is always better to add a little bit of sugar and salt rather than giving just plain water. And this is exactly the reason that it's clear. If you just have just plain water, which is like instead of a Gatorade or Powerade, why you just don't drink water? That should technically work, but it will work, but it will not work as efficiently if you have a sugar and salt in there. If you just have plain water, that's your dietary water, a little bit of it is going to get absorbed, rest is going to go through your intestine, that's it. If you just add a little bit of sugar and salt, sugar means I'm thinking glucose here, and salt, this will trigger this process to bring the water along with them as they are being transported. So that's the reason that we when make an ORS, oral rehydration solution, even in some of the conditions where you do not have the Gatorade or the Powerade in the areas where you have cholera or something like that, they just add a little bit of sugar and salt, and that's your ORS or oral rehydration solution. Works much better. The sodium is coming in and the glucose is coming inside the cell. That will bring the water to the paracellular transport. So that's basically the overview of the idea. Any question, confusion? Make sure that you get this one clearly. So if you get this one clearly, this is also similar. So here what we are seeing, that acidification of the stomach, and what is happening right here, you can see that the proton is being, right here, is being pumped in, into the lumen, and this proton gradient is, as I said, is even 1 million times, and that actually acidifies. Remember that your acid, by definition, is a proton donor. Where the proton is coming from, it is coming from the breakdown of water. So as the cell is making these protons and pumping it into the lumen, think about that, what is happening in the cell. You have this high amount of hydroxyl that is being built into the cell. So if you build up more hydroxyl into the cell, what will happen to the pH of the cell? It will go up or down. So everybody clear that you're building up hydroxyl, so the cell pH will go up, and you cannot allow that to happen. So to have that, you have this system. So the carbon dioxide diffuses into the cell from the blood, and it actually makes the bicarbonate, HCO3 minus, by binding into the OH. So it mops up that hydroxyl, and that's the reason that the pH is actually maintained, and your bicarbonate will go out back into the lumen to be transported so that's the means by which the carbon dioxide is transported into the blood. It will go towards your lungs. So that's how this hydroxyl is mopped up. Now you can see that there are some other things happening. As you are pumping out the protons into your stomach, you are exchanging it with potassium. So potassium is coming in, and this is a problem. So here you have the potassium channel, which again will leak some of the potassium, but that is not enough. See that both of them, H plus or K plus, whatever it is, these are all positively charged ions. So you will have a high amount of, you will have acidity, but you will also have high amount of positive charge towards the plasma membrane or towards into the stomach. And that's, you will not have an electroneutrality. This is a problem for cellular communication. So to maintain that, what you are trying to do, that as the bicarbonate is being pumped out, you are getting the chloride inside the cell. This is a chloride bicarbonate antiporter. So chloride is getting inside the cell. As chloride is going into the cell also do have chloride channels. As chloride is being built up into the cell, you have a chloride, high concentration of chloride, and the chloride will leak out into the lumen as well, and it will balance the charge, H plus, Cl minus. It will balance the charge of the proton so that there is an electroneutrality maintained, and your HCl is hydrochloric acid by itself. So this is how the drastic difference. Here you have a pH of 1, 7.2, and in the blood it is a little bit higher, 7.4. And that is actually maintained by the function of this. So definitely take a look at your book chapter as well. So a lot of different transporters. So you can see that for each one of them, here you have 4 transporters. Here you have 4 transporters. Just make sure that you remember and correlate the function of each. All right, so the last part that we are going to talk about, that some of the defects in ion channels, why these are so important. And just a couple of them that I just highlighted are important. One, this benign familial neuronal convulsions. It's an epileptic convulsions due to a potassium channel. Cardiac arrhythmia can happen, many different genes, potassium, sodium, calcium channel problems. Then myasthenia gravis is a muscle weakness. That's due to the sodium channels. And then you have this cystic fibrosis. That's we are going to talk about in detail. And because it is actually a huge problem in this country. I mean, like many of the different countries all throughout the world. And it's a clogging infections of the lung, and basically all small ducts are clogged with a thick mucus, and there are lots of differences. So lots of different issues that they actually, the patients actually feel due to this. And it's a recessive trait. About 1 in 20 are individual carriers. And you can see that more than 30,000 people live with the cystic fibrosis in the U.S. And that's one of the reasons that what happened, that this is the reason that I'm just putting this slide, because it was huge important in that 1989, Francis Collins, he was at the University of Michigan, and then Lapchi Siu, he was in the University of Toronto. They actually collaborated, and there was an effort to find out the gene for the cystic fibrosis, and they actually found it on chromosome 7, and there's a deletion of a phenylalanine 508. And 67% of the CFs, this gene is deleted. About 5%, you have a glycine 551 is changed to then aspartic acid or aspartate to aspartate, that also cause, and the others are something different. So, so Francis Collins, probably you know, so he is now the director of the National Institute of Health. So all the research that goes on that's funded by NIH, so he's the boss of distributing those $1 billion. So he's so he is there for a long time. So he was the director, second director of the Human Genome Project as well. So again, the CFTR, or it is called as a cystic fibrosis transmembrane conductance regulator, and also known as ABCC7. So it's an ABC transporter, expressed in many different ways, and this is how it actually works. The first thing that you want to remember before we start, I actually had a modified image of this one. By chance, if that was up for 1 or 2 days, by chance, if you have downloaded and if you see that there is a red and then yellow dots right here, just completely ignore that. That is not supposed to be there. So that's a different slide. So this is perfectly fine. This is your book image. This is perfectly fine. And the first thing is that what we want to emphasize, that most of the ABCs, or the ATP binding cassette, are pumps. So they actually transfer material from low concentration to higher concentration. This is a channel. This is a channel, and it's a gated channel. So it is not open all the time, but this will conduct the chloride ion down the concentration gradient, not up. That's why it's a channel. So how it works? You have the same way, the structures are always same. Your A1 and A2 are the ATP binding domains, also called nucleotide binding domains or NBD domains. And your T1, T2 is the transmembrane domains. And you can see that at this point, when the A1, A2, there are some sites which could be phosphorylated. They are empty and the channel is closed. So first what happens, one ATP binds to one of these domains, and these two A1, A2, these two subunits come together. That first phosphorylation to the regulatory domain is not sufficient to open the pump. So your first phosphorylation just brings them together. You need a second ATP bind to the second regulatory site, and now you have then A1 and A2 are bound tightly. That tight binding is needed to open the chloride channel. So it's a double phosphorylation is needed. So that is very important. And you can see that as long as these residues are phosphorylated, this channel is open. And this channel doesn't always close and open. It doesn't happen like this. If ATP does 2 phosphorylation, it keeps it open for most of the time. And it will be only closed when it is needed. At that time, one of the ATP will be hydrolyzed, and you can see that even at one of the ATPs hydrolyzed, they will go out, that ADP, and it will go into this form where it will close again. And again, the functioning is very simple. It's not anything important or any rocket science here. Just only 2 ATPs are needed to open the channel. That's it, and it goes down the concentration gradient. But that simple pump, it's not simple, but again, that is hugely important for the regulation of the mucus that you may develop around many of these airways. So what happens here? So what you are seeing, this is the airway in a normal lung. This is the airway lumen. And this is your CFTR that pumps the chloride, bless you, pumps the chloride from the cell into the lumen. And you can see that the paracellular transport also transports sodium in between the cell into that. So you have sodium and chloride. You have sodium chloride into the lumen. That will draw water. It can draw water from everywhere. And as you can see that any salt will attract water. And that excess water will keep this mucus light. So that is the normal thing. So the mucus will not be dense. It will be the normal normal consistency of the mucus will be maintained. What happens that if that pump is non-functional, the chloride cannot be transported into the lumen. As chloride is not being transported to the lumen, sodium is not coming in as well. So the function of the sodium is just to maintain the electroneutrality. So as chloride is being pumped, this is just like as H+ is being pumped into the stomach for acidification, it pulls the chloride to maintain the electroneutrality. This is the same thing. The chloride is being transported into the lumen. That will pull the sodium to maintain the electroneutrality of the area, but sodium chloride will bring the water as well. But if chloride doesn't go in, sodium will not go out into the lumen. There is no water transport, and this mucus is going to be extremely thick. And this is the biology of the cystic fibrosis. So remember that process. And the last one is that this will be the first topic of your module 2. And this is the bulk flow across the membrane. We know that this is known as endocytosis. So the endocytosis can be 2 types. If it's a large structure, that can be taken up by the phagocytosis, like a bacteria, spent cell. Say, for example, macrophages eat up the red blood cells. If it is a fluid droplets or tiny particles, that can be taken up by the pinocytosis. These are the 2 types of endocytosis. Or if there are some secretory material in your cell, that could be secreted by the process called exocytosis. And a classic example, we just saw the mucus. Mucus is actually a polysaccharide with some proteins associated with that. The mucus is formed as a mucin. Mucin is the polysaccharide with some protein. That mucin is actually secreted by the goblet cells, which are in your epithelial lining. They secrete the mucus the same way by the exocytosis. So the mucin goes out of the cell. If we just go into that, so the mucus goes out into the cell here, and some of these cells will be mucus producing cells. That mixes with the water to make the mucus. And here these are could be mucin secretory material. So no matter what, all these bulk flow across the membrane, whether they are coming in or they are going out of the cell, this needs a huge amount of ATP, because somebody actually asked me, Bobby was in my, we had a talk about that all of these actually need a good amount of membrane fusion. And during the membrane fusion, they need a lot of lipase activity. And due to that, it needs a huge amount of energy to do that. So we are going to talk about that in the next class. So that's pretty much end of your module 1.2. That will be all that we have for your, can you read from the back? Is good? Yeah. I uploaded this in your canvas as well, so you can see that. So let's go one at a time, what we are going to see here. So first, we are looking at that isolating cells from the tissues using physiological properties cells. So definitely know that what's your trypsin collagenase. These are your proteases that will detach along with the EDTA and calcium. know the function of the EDTA and the calcium. So what happened that these CAM or these adhesion molecules or the cell adhesion molecules known as CAMs, which help the cell to attach onto the surface of the culture vessel, like the flasks. So you need those calcium chelation. EDTA mops out all the calcium that is you have in your cell culture media. And when you do that, those cell adhesion molecule will not work. And along with that, the trypsin and collagenase will also try to separate them. So definitely know that. Then know the flow cytometry and know how to read a fax data. So we talked about that, and there were some questions about that. So remember that when you are looking at the quadrants as such, we name the quadrants A, B, C, D, or 1, 2, 3, and 4. That's how the convention. You don't have to remember that. I will provide that, but just saying that how it is. And what you will have, you have a color 1 here, and you will have a color 2 here. So you do not have to memorize any name of any colors or any fluorochrome, whether it is red or green, or what specific antibody is conjugated to what. You do not need those information. Those will be provided. But if we are looking at a certain type of thing, that if you have something here, what you may conclude depending upon the questions, or if you just have something here, what that conclusion that you can draw from there. So definitely try to understand this type of problem. So you will have, so all the questions will be multiple choice, but there will be figures in the question. So just make sure that you understand that. It's not wording. So several places you could have figures. Cell culture, nutrient requirements, just an overall, they need the amino acid and the vitamins. They need fetal bovine serum as a source of growth factor. That's important. So definitely need that you need the serum, growth factor. That's the source. Transformed cells, and then transformed cells know that they are weird in the sense that they are aneuploid, high density attachment requirements, and they can differentiate into multiple types. So that's one of the problem, that when you are working with the cell culture, no matter whether it's a human cell or anything, it may not mimic a cell or the environment which is really inside what is happening in a physiological condition inside a human or a mice or a rabbit's body as such. So that's the reason that sometimes, because all these transformed cells have compromised genetics and biochemistry, that's the reason that sometimes it is not always apples to apples or orange to orange comparison. That's a problem. So know the hybridoma technology. That's definitely know that the protein that you are trying to raise the antibody against, that needed to be injected to the mouse. That's very specific. And then you isolate those cells, you fuse them with the myeloma cells, and then you use the selection media. And know that why the selection media is important and how cleverly you can select the cells that will only produce the specific antibodies, and you can isolate that. So definitely know that. Pharmacological methods, I just named them. Again, I don't expect you to remember that whole big chart. So these are just a couple of them. See your brafaldin A, cytocalysin, nopudazole, Golgi function, disruption of microtubules. PMSF, I said in two different areas, that phenylmethyl sulfonyl fluoride. That is a protease inhibitor. That always, so we are going to come back to that. So why I said that is very important, that whenever you are isolating tissues or anything like that to isolate a protein, isolate an organelle, as soon as you try to break up the tissue, we'll come to that, which you do use sonication or disruption. Before you do that, you always have to chill the cells and add this material so that the protease inhibitors are, this is a protease inhibitor, so that the proteases which will, as soon as you disrupt the cell, all these proteases will be on the loose. And whatever they find, these are all serine proteases, they will chop at arginine and lysine residues so that this actually doesn't happen. You need a protease inhibitor in almost every experiment in cell biology when you are isolating this material. After the purification, when the protease is gone, you don't need that. But at the initial step, that's why it's very important. All right. So long list. So let's take a look at the whole cell techniques. And again, some of these are very straightforward. Just know that what it is. I will really not go in between the lines in the experimental part because you are not, a lot of you asked me that how much detail on those. As you are not working with these with your own hand, for the experimental technique, we will go more of a superficial idea about the concept rather than just going in between lines into the procedure because it is not fair in that sense. So just the definition of magnification, resolution, know the steps of sample preparation, that how you fix them, then if it's needed to be embedded, you embed, section, stain, those are the processes. Bright field, phase contrast, DIC, what's the basic difference? You just do not have to go, someone asked me that whether you need to know where is your sample, then you have a condenser, then you have a stage, then you have objective, then you have projection lens, or instead of that dichroic mirror, or you do not have to remember what is after that. You do not have to. That will be too mean to do that. So yeah, just the basic difference. So the phase contrast will have these annular plates, which are called the phase plates. So remember that. Nomarsky interference will have these Nomarsky prisms, which actually make this difference in the three-dimensional image. Just the main thing. Nomarsky prism in the DIC, whereas the phase contrast will have the annular ring or the phase plates. So usefulness, each one of them. And then, say for an example, we have just a couple of examples of that hematoxylin eosin stain or other type of stains. So suppose a typical question is that you have a sample, a biopsy sample from a patient, and you take the sample, you fix that, you stain that, and you are going to look at the morphology of the cell, arrangement of the cell. What kind of microscopy will you use? So you can use just simple bright field microscopy if it is, so your main important part is that once you actually fix the specimen, immediately you know that it is not going to be DIC. DIC, you always look at live cells. Phase contrast, also you look at live whole cells. Whereas the bright field microscopy, as soon as you fix it, section it, and stain it, you will use the regular light microscopy to do that. Now, if you are looking at the overall morphology of the cell and the arrangement of the cell into the tissue, light microscopy is good enough. If I ask that you are looking at the structure of the lysosome or peroxisome inside the cell, that will not be light microscopy. You have to go into the electron microscopy. So these are some of the concepts. Just make sure that what I'm asking for, overall cell morphology and the tissue structure, light microscopy is good enough. So staining general, then fluorescence, important that there could be some mean question. Principles of excitation and emission. And the emission wavelength is always, ignore that, equal, is always higher than the excitation wavelength. So if you have an excitation wavelength at 500 nanometer, I'm just saying arbitrary 500 nanometer, your emission wavelength cannot be 490. That's a very important. It can be 510, but not 490. Emission wavelength is always higher than the excitation wavelength, so be careful about trick questions about that. So emission, excitation lower, emission higher, always. Then immunofluorescence microscopy. Know the principle, and here, just make sure that you have a very clear idea about the primary and the secondary antibody. The way to visualize is that first you have your protein, then you have a primary antibody. To visualize this protein and the primary antibody, you need a secondary antibody to which you have a fluorophore attached. And the second important part is that whatever that primary antibody is raised into, your secondary antibody will be anti that antibody. So your primary antibody is mice, your secondary antibody will have to have anti-mice. If you have a rabbit, your secondary antibody will have to have anti-rabbit. So that concept, remember that. You do not have to remember the name of rhodamine, fluorescein, which is green, which is red. You do not have to remember any of those at all. Live cell imaging, confocal microscopy, mainly the confocal and deconvolution microscopy. Just know the advantage that you are getting over your fluorescence. The main difference for the confocal is that you are using just only one specific focal length, and you are allowing the light from a very specific focal plane to enter your projection lens. So what you can do that when you will actually study a confocal image, you have a focal plane here, you have a focal plane here, you can study each focal plane separately and build a three-dimensional image, which will be much clearer. When you have a fluorescence image, you have a focal plane of this much thickness. All the light from all these focal planes, where it is here, 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 all the focal planes are getting the lights into your projection lens. So which is much more inconvenient, and that's why you get these blurred images. So on the other hand, this is an easy way to remember that somebody told me, is like confocal means slices. So you can look at slices of your material, and that's the most important. You can focus your confocal beam to each slice. So that's the idea. Then fret, remember the principle that you have two fluorophores which are attached to the two proteins, and these two proteins need to be very close, usually in the range of 10 nanometers. If they come that close, the fluorophores, if they are designed that way, that the first fluorescence molecule or the fluorophore's emission wavelength is near or equal to the excitation wavelength of the second molecule, you get the excitation of the second molecule. So that's the principle. And then just remember that how you can, we did this in the class. So just remember that we can ask some question in that sense, where you have a list of fluorophores, and you have to pick up the pairs. Now remember that it could be just a little bit different, and again, instead of just writing the wavelength, we can just give you an emission, sorry, not like that. We can give you a chart like this, and you can have some numbers in here. So just, which will be clearly written, say for an example, this is 450, this is 500, this is say 600. So instead of just a chart, you may have 3 or 4 tiny of these where you have the wavelength, and you have to just kind of find out what is the wavelength of the excitation or the emission spectra. So that could be the differences as such. And again, it is not that you need a microscopic thing. It's just will be very, very obvious type material. So again, for FRAP, the same thing. Just to know the principle. We're going to talk about that in the next part as well. Iron sensitive dye, we just talked about just only one. There are many other, but just a Fura-2 with the calcium. So remember that. That's a kind of a remembering questions that it can do that. Super resolution microscopy, and just the basic principle in just what each one of them do. Advantage over confocal over a regular fluorescence microscope. That's one of the things. And I was telling a couple of people who stopped by during the office hours, all these experimental techniques I worked with my hand like for years, like including all types of electron, including cryo-electron as such. So I know those like the back of my hands, but I did not work anything with a super resolution. That is complete. Your knowledge in the bookish knowledge, my knowledge is exactly the same. So because, yeah, I mean like, no, that just be real. I mean like I did not, I have not been to a facility where I could work with a super resolution because it is just so recent. So if anyone is interested, I can just post that. February 20th, there will be a lecture at the genetics biochemistry. they are going to discuss about the STED, super resolution. Leica is actually coming in. They probably may bring a machine, but they're going to be a seminar. It's open. You do not have to do any registration or anything like that. So if you are interested in that, you want to go to graduate school, just go and look at, you can just attend that seminar. So again, super resolution, just the basic thing that how they work. That's good enough. Then transmission, just know the principle. That's what the advantage of that. Then negative staining and metal shadowing. Negative staining, know the stain names like osmium tetroxide, uranyl acetate, which phosphotungstic acid. These are all heavy metal, and the principle that these are make the electron dense, dense condition that will deflect the electrons and make a contrast. So that's very important. This was one of the questions that I think that someone asked me during my office hours, and I think that I should share that as well. That if you actually look at the picture of the metal shadowing, which was just a few poliovirus picture, and the question was why you were not doing metal shadowing and using an ACM. That was one of the question, and why you don't routinely do this with the transmission electron microscopy. So the first thing is that your metal shadowing technique with using the transmission electron microscopy is probably 1% of the whole technique that you use. Most of the time that the transmission electron microscopic technique that you use, you just take a direct virus, you negative stain it, you visualize that. You will see that there's some rotavirus, which a very common picture that you see. It is not three-dimensional, it's just two-dimensional picture of a virus. That's the common transmission electron micrograph. Or what you do, you see a section of a cell, like an onion root cell, a bacteria eating phagocytose, a macrophage phagocytosing some bacteria. These are all section specimen, just black and white. These are your standard transmission electron micrograph. Now, why you do the metal shadowing and what are the problems for this? So the metal shadowing gives you a three-dimensional image. Now, the question was that why you cannot do that with the ACM. Now, remember that right here, if you look at that, the scanning electron microscope actually have a considerably lower resolution compared to electron microscope. This is just for your understanding. I don't think that I'm going to ask these questions. This is just an overview of that. Lower resolution. So the picture of that metal shadowing that you have is a poliovirus, which is 30 nanometer in diameter. That is too small to be visualized with a scanning electron microscope. You cannot visualize that properly. That's the reason you actually are doing the transmission electron microscope. On the other hand, if you just need to look at the surface of a cell in a 3D image, you can easily go to scanning electron microscope directly. You have a picture of that, some epithelial cells looking the cilia and all those kind of things. You can do that because they are much, much larger. You have that option. So those are some of the reasons that you do the metal shadowing. But most of the time, negative staining of viruses is a direct stain. You take the virus, you stain it and visualize this. If it's a thicker specimen, you slice it, stain it, and then visualize it. These are the techniques. Now what happens that, I'm skipping the immune and come back. Cryo-EM, remember that cryo-EM is a transmission electron microscope, extremely powerful. Usually it's about, so what we mean by powerful, that it depends upon the intensity of the electron beams that is ejected by an electron microscope. Usually a transmission electron microscope start about 70 kilovolt of current put into the tungsten beam so that it heats up and puts the electron beam. It goes up to 150, 200 kV, kilovolt, means 200,000 volt, that's it. So for a cryo-electron, it goes up to approximately, the range is about 300,000 or 300 kV. So it's a very powerful, so the penetrating power is much more. And why you cannot have a high penetrating power and what is the pros and cons? This is something extra I'm not going to ask. If you actually have a very intense electron beam, it's going to destroy your samples as well. So in the transmission electron microscope, you have this copper grid. On that you have your sample, and if you're just too much intense electron beam can actually evaporate your sample either. So that's a problem. On the other hand, what happens, cryo-EM, you can utilize that high beam because it is in a liquid nitrogen temperature in vitreous ice, and that cold temperature will actually allow the sample to have its integrity. So not evaporating the sample. So that's the reason that you can use that strong beam of electrons. So again, the main principle, remember that, and the advantage over the regular EM for the cryo-EM, you do not have to do any kind of staining or fixation. You take your sample and immediately you freeze it in just water with the liquid nitrogen temperature, and it will form the vitreous amorphous ice, glass-like ice, no crystals, and that is good enough. And you just tilt the specimen in different orientation, and your computation power is the main thing that helps you to build that material. On the other hand, what you can do that when you do your regular transmission electron micrograph, you still can make three-dimensional images, but you have to fix your specimen. After that, you have to stain your specimen. You have to go to, with the microtome, you have to slice, and you have to keep track of the slices, and then you build the image on top of that. So you have this problem of that whole technique is much more laborious. So you have the picture of the Golgi in a three-dimensional reconstruction, where it's an extremely laborious process to do all these technique before you get that whole thing. On the other hand, you take a whole cell and you can just do the cryo-EM, and the penetrating power will allow that three-dimensional image to be taken off the Golgi out of an intact cell, and you can build that much faster without that whole preparations process. That's the advantage of your cryo-EM over TEM. All right, immune-EM. So immune-EM, again, if you need to localize, pinpoint a specific protein or an enzyme, which is a protein, then you can use that technique. And again, remember that no matter what, wherever you are trying to do an immunofluorescence or immunoelectron microscopy, and if you are using an antibody to detect that, you always have to kill and fix the cell. That is very important. You cannot do that imaging on a live image. Of course, you cannot do that EM, but even for fluorescence or confocal, a live cell will not take up an antibody. Antibody is a large protein molecule, hydrophilic glycoproteins. Antibody will not be able to enter a live cell. You have to fix that and permeabilize the membrane. Usually you use ether or acetone to permeabilize the membrane, so a part of the membrane is disrupted, and then you can use the antibody so that it can go inside the cell and bind there. So again, the protein three-dimensional structure is maintained. That's not a problem, but you always have to have a fixed cell to do immunoelectron or immunofluorescence microscopy. And for the immunoelectron, you can just attach a gold particle. There are two ways. You can use a secondary antibody, or you can buy the protein A attached gold, which is much cheaper. So you just buy them in a vial. You have this gold particle, different sizes, 5, 10, 15 nanometer, and they will be attached with this protein A from Staphylococcus, which is commercially available. So all you have to do is that have your primary antibody. It goes and binds to the specific protein. You add the protein A will go and bind to the Fc region of the antibody. So if you remember that you have the Y-shaped molecule, and this is your Fc, your protein A will bind to that automatically, which is attached to the gold. And that you can visualize with that. So on the other hand, you can always do that secondary antibody as well. So this is your primary, this is your secondary. That's the normal way that the fluorescence or immunofluorescence, you can do that in both conditions as well. So next we are looking at the disruption of the cell. And one of the way that you do is the sonication. So we had a conversation with this. Suppose you have a piece of meat. You want to extract some mitochondria out of a steak or some chicken. So how you start, I mean, like literally how you start. You take those meat and you will actually blend them just initially with just a regular kitchen blender. You have to keep them in cold condition. You blend them, and then at that point you have to add those protease inhibitor, or otherwise they're going to chew up your cell. You need your PMSFs. And then what you do that after you actually do have a good amount of homogenate, a good number of cells, they are not individual cells, but tiny small number of cells. So you have a kind of a slurry of cells. Those slurry of cells you take and do the sonication. So the sonication, what you can just put it in a beaker or in a test tube, and you have a probe which is just which could be really tiny thin depending upon the volume, and you just put that probe and it vibrates at the ultrasonic vibration. So that ultrasonic vibration is going to actually disrupt the cells. So the cells are going to burst at that point. So all the cellular material will come out, and then you do a low speed centrifugation so that all the cellular debris will settle down, and then you can do the velocity centrifugation. And you know that each step is 600 G, your nuclei will precipitate. So each time what you do that when you do the velocity centrifugation or anything, so you will spin it with a certain G, and again this G is that the gravitational force. It's a relative centrifugal force that is actually felt by that. So again, I did not go into the detail of those techniques, but just have an idea that you have a rotor, which could be a fixed angle. You have a rotor here, you have a rotor here. And you are spinning that. As you are spinning, you are having a centrifugal force working in here, and your material will precipitate. So this is what is called a fixed angle rotor. There are some which will actually swing out, so the rotor will be like this. So these are some buckets right here with a hinge, and they will just fly out and swing out, and nothing will come out. And those will precipitate at the very bottom, just not on the side, depending upon what you are doing with those. So these are the different way. And again, you spin, and the material that is being spun will actually have a relative centrifugal force acting on them. And how it actually depends, if you are close to the axis of the rotor, you will have what you call as the G will be minimum. Then here it is a G average, and here you will have a G max. So think about that when you ride those spinning rides in your county fair. If you are sitting outside, you actually have a more G and you are like this. Whereas if you're just closer to the axis, you feel a little bit less G force. It's the same way you are precipitating your material down, and that's how it works. So what happens that after each step of centrifugation, you will have a pellet right here. And this is what is called the soup or supernatant. That is your pellet. And suppose at 600 G, in your pellet, all your nuclei is in the pellet. And you take out the supernatant, everything else in the supernatant, and you put it to the next one. And the next one will again have a pellet, and then you just take the supernatant and put the next one, and this is how it works. So this is the first way that how your differential velocity will separate the material. Now, if you have a similar material, like your chloroplast, mitochondria, lysosome, peroxisomes, they almost have a similar density. Then what you need to do that from there, you take the supernatant and you have to put that on a gradient. And this gradient that we talked about could be sucrose, cesium chloride, many of those. And then you do the centrifugation. What happens that they will try to move down, and they will be stuck where the concentration of the gradient matches the concentration of the material. So you can have in a fraction your mitochondria, chloroplast, or the lysosomes in different areas. You puncture the bottom of the tube, and you collect the material from there. So that's basically what it is. Here I just added something that how will you separate two same density vesicles? Sorry about that spelling mistake. I will just correct that. Two same density vesicles with a different protein on their surface. So what you can do, this is an experiment that is described in your book. You have a picture of that. You can easily add an antibody, and you can attach a protein A or anything like that to precipitate that, and you can isolate that. So this is your affinity purification. You can do that. Mass spec, just know the principle. That's good enough, that how you can just do that. Again, this is a separate chapter with just that you can identify some of the peptides or protein that you isolated from an organelle using a mass spec very, very quickly. All right, so after that we can go into the looking at the genetic method. And again, for the genetic method, if it's needed, it can be gone on into very detail. But as you are not doing these experiments with your own hand, we will not go into the in-between lines procedure as such. Like, I will not give you a germline that this is a germline which you have an X minus, and here you have a small a, capital X minus and X plus, and you find out that how you can do that. You do not have to worry about those things. We are not going to ask. So again, just the take-home message that what you are doing. So when you are trying to do it at the genome level, you can do a germline knockout, or using a CRISPR-Cas9, you can actually disrupt the gene function as well. So this is at the genome level. You can use these two techniques. On the other hand, you can do this Cree and the LOXP, and the advantage is that what you can do, that the Cree, you can put a promoter just in front of that, which is tissue specific. So the Cree will be only active in liver cells, suppose. And you want to just only knock out that gene in the liver, but not the rest part of the body. Or if you just want to knock out a gene in the brain or the nervous tissue, then you have a neuron-specific promoter in front of the Cree. So that's how you can specifically knock out a gene in a tissue. Then level of the protein, expressing a dominant negative protein. So again, just an overview of that. Suppose here the example that we have, that you have a GTPase. So for the GTPase, the function of the GTPase, you do not want to touch that. But for it to function, it needs another protein, which is the exchange factor, with which it can change that GDP so that its site is empty so that another GDP can bind. And you can alter the function of that other protein that is needed in the life cycle of the protein, or the recycling of the protein, or the functioning of the protein. You can mutate that, and you can knock out the protein function. So this is your dominant negative mutant proteins, or the dominant, or shutting down the dominant negative allele. And then at the level of RNAi is how you easily can disrupt the gene function at the mRNA level, designing that specific type of mRNA which will form by the dicer, the double-stranded RNA. And again, some small things that remember that what the two differences. Initially, that RNA is formed as a hairpin. Everybody remembers that? This hairpin is removed by whom? Who does that? Yes. So remember that that's just the basic function that the dicer will remove and just make it just a double strand, 23 nucleotide double strand like that. And then the RISC will load that. So this double strand will become single strand, because the double strand cannot load onto your mRNA. Your mRNA is a single strand. So remember that from here you have your mRNA, and this will be now becoming this. So here this loading of this portion, where this removal of this one strand and to single-stranded RNA and loading onto that mRNA, this is done by whom? The next one other than the dicer? The RISC, the RNA-induced silencing complex, which have the other proteins, the RISC will do that. So these are the things. I mean, like, I'm not going to ask you in between do that. And how you study localization, we already talked about that. That's good. And Let's take a look at the function of membrane. So definitely know all the phosphatidylcholine, serine, ethanolamine, what they are, which are neutral and which are charged. So definitely know that. Know the difference between the sphingolipid and the differences. Now remember that the sphingolipid could have two different types of head groups. So remember that you can change from one head group to the other, and make sure that you remember those. It is not a phosphoprotein. It's not a phospholipid. It's a different class of lipid. So definitely remember that. Also know the cholesterol also do have a polar head group, and this polar head group comes from a hydroxyl group. So that's important. That definitely remember that the hydroxyl group is the polar head group. Then you have a planar ring and a tail, and all of that is hydrophobic. And so know those things. Bilayer versus monolayer, remember that you the bilayer, you have the two separate ones, whereas the middle portion is covalently joined in a monolayer, but each end does have a polar head group. So know that. Lipids are fluids and the movement of the lipids. So when we actually change the transition temperature, here it's an important thing that know the principle and analysis of a given set of lipids with a different chain length and unsaturation and prediction of the transition temperature. So for an example, if we just give something like this, that if you have a fatty acid with 14 carbon, then you have another fatty acid with a 14 carbon with 1 saturation. Then you have another fatty acid which is 15 carbon with 2 saturation. Got it? So I'm just writing it down right here. So 14C, no saturation, is like complete saturated. So this is a 14 carbon fatty acid, completely saturated. No double bonds. Then you have a 14C, 1 unsaturation. Means 1 double bond. Then you have a 15C right here with 2 unsaturation. Everybody good? So now if we just try to make temperature differences, like a hypothetical temperature differences for their transition temperature for these, I'm just putting it something like this, like random 56, 55 degrees. Then I have something like 5 degrees, and then I have, say, minus 10 degrees. So if I have these numbers, where will you put it? Where do I put the 55? It's just random number. You do not have to memorize any of these. And then 5. And then minus 10. So everybody good? That how does that work? And again, remember that. Do not memorize those numbers from those charts. Those just have an overview of idea, because these will be drastically different. All you have to do that just one saturation will drastically reduce the numbers compared to the chain length. And if you get the principle, you will get this. So just that idea about the transition temperature, what the different transition temperature can do. So movement of the lipids, and again, also remember that the chain length and the unsaturation, those are the main things that's important, and the thickness of the membrane changes with the temperature. Asymmetry. Yeah, membrane asymmetry, there are some of them which are like a little bit carved, conical shaped. Those can change the membrane curvature, know those. And integral membrane proteins, transmembrane, lipid anchor, and peripheral proteins, know those. And here the most important part is that we talked about that when we actually use the detergent, how do you isolate that integral peripheral proteins with the detergent? So that's actually the important part that is important. So remember that all your peripheral proteins are attached to each other by mainly with weak electrostatic interaction. So when you have this electrostatic interaction, or at the most van der Waals, which are very, very low energy, you just can add some salt and like a sodium chloride, and your peripheral proteins will come out. All you need is that's it. Your peripheral proteins will come out. There's no problem with that. So again, repeating, your peripheral protein to extract from some membrane, salt is good enough. Whereas if you have an integral protein, you need a drastic change. You need some detergent like SDS or Triton X100. You need a detergent to extract your membrane integral proteins. So everybody good with that? You need a detergent for that. Now, if it is a lipid anchored protein, what will you do? That's a tough one. If it's a GPI anchored protein, how will you, because okay, all right, you isolated it from the membrane, but still a lipid is covalently attached to the protein. You cannot get rid of that lipid. Lipid is a lipid anchored. I will not ask that, but still what you can think that could happen. Anyone? We talked about something about that breaks in membrane integrity present in snake venom. That can give you idea. Phospholipase. You need an enzymatic treatment, phospholipase, to break those lipid and protein interactions. So for the GPI anchored protein, you need phospholipase, enzymatic digestion of the protein to separate it out. So again, repeating, peripheral protein, salt is good enough. Integral protein, you need a detergent like SDS, sodium dodecyl sulfate. For GPI anchored proteins, you need a phospholipase. All right, so what else? Then how to read a FRAP data. So for an example, if I give you a curve, like initial FRAP was like this, and then it goes up like this. So this is the point where you are bleaching the membrane, and after bleaching you got this one. So here, if we just read that, is the protein mobile or immobile? Mobile, how many people mobile? Good, okay. So everybody clear? It's mobile because it's recovering. If it is like this, then is it mobile or immobile? So everybody good with that? It is not recovering from the bleach. That's the reason that is immobile. So just make sure that this is basically how you can read these data. All right. Then just epithelial cells, like they can separate the proteins using tight junctions. Know those. Lipid rafts, they can localize different proteins using those specific ones. Know that what are the specific lipids that is present in the lipid raft. I think that's pretty much what it is. All right, so here, facility diffusion, active transport, uniporters. Yeah, so here, mainly straightforward questions, but know the GLUT1 and GLUT2 difference distribution analysis. Definitely read that. Again, if you just look at your book, you will see that there is a huge amount of enzyme kinetics. You do not have to do that much of mathematics at all. So the only at the most, and you may find this question in your quiz, so you could be better prepared. So suppose you have a 10 millimolar glucose and a 100 millimolar glucose, and then you are just given a Km of one protein is 1 and another is 10. So if it is given something like that, you try to figure out that which is going to pick up glucose faster at a certain concentration or anything like that. So again, you will find this question, I'm sure, that in your practice quiz. So definitely take some time to do that one, and those kind of things will help you. So the different pumps, classes, function, opposite nature of V and F class pumps. Remember that both of them are proton pumps, but there are two different ways. For your one of them, pump the protons in the expenditure of ATP, V-class pumps, where the F-class pump, like in F1 ATPase, it actually pumps the H plus from high to low to generate ATP. So remember that V-class pump and F-class pump the protons to generate ATP, whereas the V-class pump use the ATP to pump protons. Main difference. Physiology of congestive heart failure. Definitely remember the sodium-potassium pump, the sodium, and along with that, the sodium-calcium antiporter. Definitely remember that. Digoxin, aubine. ABC transporters. What happens in the MDR, or the multi-drug resistance overexpression in the cancer cells. Know those. Transepithelials, we spend a good amount of time, so definitely have a clear idea that each one of them have 4 transporters and how they work. GLUT2 instead of GLUT1 in the intestine. Definitely remember that. And also know that your cystic fibrosis, the CFTR is a channel, not a pump. And I think that's it. So along with this, what you're going to do, definitely still take a look at your clicker questions, and still even your quiz will close on Sunday, but still do the practice until even before your exam. So that will help you out. You will see some similar question from your practices. All right. Yeah.
Did you do this today at all? It wouldn't let me like join.
No. I didn't run it. So yeah. Those are some of the buy days. Yeah. So the days I take attendance, it will be the 5% of those days. So you can easily see that somebody even without that will miss that. Yeah, question? What? So initially I thought 50 for 75 minutes. I have to just have a look. Either 40 or 50. If it is 40, it's 1.25, like 2.5, but I think it's 50 questions, 75 minutes, yeah. Yeah.
On Monday, you got any time for an appointment?
Yeah. Anytime. Yeah. Absolutely. Yeah, question?
I need to head home this weekend. I just had a family member hospitalized.
Okay.